Hello, everyone, and welcome back. We are going to start a unit on evolution. I have a really interesting video for you to watch about ice fish and how they have clear blood. It's going to really play into something that we're learning about today. We are going to be on page 213 in your binder. So you're going to want to flip to page 213 in your binder. This is chapter 18, and we're going to jump from genetics into evolution. It's a really nice, learning about genetics is a really nice segue into evolution because evolution is going to act on genetic diversity, basically. We'll talk about that a little bit more later. Chapter 18 is going to cover genomes and their evolution, kind of just a really big picture about how genomes can be altered over time and species can evolve. And then we're going to look at this topic called bioinformatics. And then the next time I see you next week, we will use a government database, and I'll show you exactly how bioinformatics is used as it applies to evolution and trying to figure out the relationships, the evolutionary relationships among different species. So pretty cool stuff, pretty interesting stuff. So you should be on page 213 now. Just to kick it off, I have this video to show you. It's called The Birth and Death of Genes, or it's called The Making of the Fittest, The Birth and Death of Genes, and it's about ice fish. And so what you're going to want to do is flip. So I'm showing you this at the beginning. I normally show it at the end of class or in the middle of class. You're going to flip to page 218. And you have two questions to answer. What is the main point of the video? And what do you find most interesting? So that's on your notes. Main point of the video, what you find most interesting. And then along the way, just to make sure everyone is paying attention, you're engaged in it, I'll pop in some questions as it goes. So I hope you enjoy this video.
For life to survive, it must adapt and readapt to an ever-changing Earth. New traits and capabilities must be invented, while some traits may be lost. To see one of the most dramatic examples of how this occurs, we will follow a voyage of long ago that will take us 1,600 miles off the southern tip of Africa to one of the most remote places on the planet, Bouvet Island. A speck at the edge of the Antarctic, it's pummeled by water so wild that getting there was no sure bet, especially in 1927 when this Norwegian steamship left port.
It's cold, it's windy, it's rough waters, they're far away from civilization. So it was a really long and arduous voyage from Norway to eventually the shores of Bouvet Island.
Norway had dispatched the expedition to claim the island as an outpost for its whalers. And after 2 months at sea, on December 1st, the crew finally rowed ashore and erected the Norwegian flag. But that's not the most important achievement of this expedition.
One aspect of their voyage was to understand all the sea life that was there. And so there was a zoologist on board, Ditlev Rustad. And Ditlev's job was to really just throw a net over the side and trawl up, see what he found. But one day, in fact, the day after Christmas, 1927, he pulls up a really unusual looking fish.
A creature that many years later would illuminate our understanding of how evolution works. A creature so unexpected, it is still barely believable today when taken from a freezer at Northeastern University by a biologist who spent much of his career trying to make sense of it.
Well, Sean, it's easy to see why the fish was originally called a crocodile fish. Notice very big protruding jaw with lots of teeth.
Incredible, but it's so translucent, it seems like I can see inside the fish.
Well, the reason you can see inside the fish is that this is a scaleless fish. And in fact, if you...
What is that?
Well, that's the brain. You can actually see the brain through the skull. And then you can also see, projecting from the brain, the optic nerves going over to the eyes. And Sean, for comparison, we can take a look at a cod. And when I pull back the gills of the crocodile fish,
Creamy white, look at that.
and here, if we take a look at the cod,
Oh, brilliant red.
yeah, very, very red.
So that would have surprised Rushdad, because all the other fish he had seen would have red gills.
Absolutely.
But that wasn't the most incredible thing about Rushdad's find. That came to light when Rushdad, like any good Norwegian confronted with a strange fish, filleted, as I'm doing today under Bill Dietrich's watchful eye.
Unbelievable. Everything's white, everything's colorless.
Yep.
All the organs, all the soft tissues, the heart even. Every other fish he had opened in his life was full of red blood, but not this fish. And he wrote in his notebook, boldly,
Colorless blood. Bleu? Farvelist. The crocodile fish, or ice fish, as they've come to be known, had a dilute liquid almost as clear as ice water flowing through its veins.
okay,
And here's your ice fish blood.
that's amazing.
Except for the ice fish, all of the vertebrates on Earth, reptiles, birds, mammals, and all other fish, like this cod, have red blood coursing through their veins.
So what's so incredible about having no red blood cells at all is that, for example, our blood, 45% of our blood by volume is red blood cells. So we have enormous numbers of red blood cells in our blood, as do most other backboned animals. And when our red blood cell count decreases a little bit, we call that anemia. And if it decreases a lot, that's life-threatening. So it's stunning that an animal could get by with no red blood cells at all.
Why would a fish abandon a way of life that has nourished its ancestors for 500 million years? It has to do with the extreme habitat in which it lives.
The more cells that are present in blood, the thicker it becomes at cold temperatures.
So this watery blood allows the fish to live in really cold temperatures. So if it had blood like this, it would be all gummed up.
That's correct.
All right.
Blood is red because it contains hemoglobin, the protein that delivers oxygen throughout the body. Yet the ice fish have not one hemoglobin molecule. The fish absorb enough oxygen from the sea through their scaleless skin so that they can make do without hemoglobin. But the lack of red blood cells still doesn't fully explain how any blood flows in oceans this cold, below the freezing point of fresh water and almost at the freezing point of seawater.
And fish cannot live generally in that sort of environment because their freezing point of the blood is higher than the freezing point of seawater.
Yet fish thrive in these waters, ice fish and other species, all members of a group called the Notothenioids. To show just how different Antarctic Notothenioids are from other fish, husband and wife biologists Arthur DeVries and Christina Chang dunk one in water as icy as the ocean outside their lab on Antarctica's McMurdo Sound. The temperature, minus 1.8 degrees Celsius, is so cold it's below the freezing point of fresh water and blood.
So as you can see, this fish is doing fine at this freezing temperature in the midst of ice crystals.
How these fish thrive in such cold waters baffled scientists until the 1960s, when Art DeVries found that Antarctic fish had invented something that protects them from freezing: antifreeze.
These fish have a certain protein, and what it does is bind to it, the ice crystal in the fish. And by binding to it and preventing it from growing any bigger, the fish doesn't freeze.
So only one group of fish in the Southern Ocean, the Notothenioids, ice fish belong to that group, have antifreeze. So it's an invention unique to this group. And that's allowed those fish to invade a space that other fish couldn't live in. And these are nutrient-rich waters full of lots of food. So these fish are thriving now where other fish couldn't thrive.
Antifreeze proteins give notothenioids a clear edge where they live. But their existence poses an evolutionary mystery. When and how was antifreeze invented? The waters around Antarctica were once a temperate 10 degrees Celsius. But 34 million years ago, Drake Passage opened as Antarctica broke away from South America. Now a continuous current circled the new continent, isolating its surrounding waters. As a result, those waters chilled to minus 1.8 degrees Celsius. So notothenioids and antifreeze evolved in the last 34 million years. But traits are encoded by genes. Where did antifreeze genes come from? De Vries, Cheng, and Lingbao Chen worked out part of the mystery in the 1990s by noticing that parts of the antifreeze gene, labeled in red, strongly resembled a different gene, that one gene gave rise to the other. The process began when the ancestral gene was accidentally duplicated. While one copy remained the same, the other accumulated mutations that eventually gave it a new function, to make antifreeze proteins.
The invention of antifreeze is a crystal clear example of inventing something new from something old, borrowing sort of the code of a pre-existing gene and then altering that to create a protein that has entirely new functions. And this story we see again and again in evolution, inventing something new from the old.
Notothenioids invented antifreeze genes, but one family, the ice fish, went a step further and eliminated red blood cells and hemoglobin altogether. As for how that happened, the answer has also been preserved in DNA. And Bill Dietrich found it when he compared globin genes from ice fish and other fish. Globin genes encode the hemoglobin protein.
So what do we see here?
Well, as you go from left to right here, the normal red-blooded globin gene is shown in the sequence on the top. Down below, we have the sequence of the ice fish. And as we move from the left, you can see their dots. These are exact matches in the sequence. However, at the arrow, we see we get virtually no match. This is essentially genetic gibberish.
So it's broken right there.
That is exactly right.
At some point in history, a mutation wrecked this gene. And since hemoglobin was no longer necessary to ice fish, the mutation wasn't weeded out.
And this is the fossilized remnants of that gene.
So if the gene has become useless, it will eventually be lost as these mutations pile up and just erase what used to be there.
That is exactly right.
So genes are born and genes die as species find ways to survive in an ever-changing world.
This remarkable fish that Ditlev Rørsted dredged up 80 years ago has taught us is that evolution doesn't always come up with the best solution imaginable. It just comes up with the best solution available. And that sometimes means getting rid of things that worked in our ancestors, as well as inventing new things like the antifreeze. And that record of changing habitats and changing lifestyles and changing genes is etched in the DNA record of life.
Okay, so there's a couple of things I just want to point out from that video. It's a great video. I really like that. I think it's very interesting. And so I hope that you did too. So the main point of the video is basically, I mean, there's a lot of points that they make, Um, but you have this fish, and the fish has clear blood, scaleless skin, and an antifreeze protein. The main point is that over time, genes by random chance will change. If the change in a gene leads to something that is beneficial to that organism will survive, reproduce, and pass on that new gene. That's basically what natural selection is. There's a couple of things I want to point out. Some of the language in the video bothers me a little bit because the way the guys talk, it makes it sound like the fish chose to have that mutation, and they kind of chose and they decided to knock out the hemoglobin gene. And I want to just make sure that I address that from the very beginning, that is a misconception, and that's something that I see students really struggle with. An organism does not choose to evolve. It doesn't choose to have genetic changes. Genetic changes happen by random chance. And the other thing is that there wasn't a fish that had hemoglobin and then all of a sudden the gene switched off and it didn't. There was one fish that had hemoglobin, gave birth to a fish that did not have hemoglobin, or several that did not have hemoglobin because of a mutation, and then those fish ended up surviving and reproducing. So individuals do not evolve, populations evolve, but we'll get to a bunch of that. So we have some notes to take. I'm on page 213. Aw, how cute is that? Another misconception I want to just make sure that I address right off the bat is, listen very closely, humans did not evolve from chimpanzees. Chimpanzees did not evolve from humans. Humans and chimpanzees each evolved from one common ancestor. And you're like, what is this common ancestor? Well, it was a thing. It was probably kind of hairy. It looked like both. I mean, that's all I could say. So if you can imagine, like, you see where my cursor is, like a little point right here, this is a common ancestor. And then if you're drawing like a phylogenetic tree, and I'll do this for you, you probably can't understand what I'm saying right now, but there was one common ancestor, and then through time, the chimpanzee and the human each evolved in their own direction. When we do phylogenetic trees, you'll understand that. I'm getting a little ahead of myself here. What I want to focus on today is bioinformatics. And what bioinformatics is, we're going to talk a little bit about prokaryotes versus eukaryote genomes, transposable elements, evolutionary development, also known as evo-devo, and then I'm just going to kind of jump to homeotic genes and homeoboxes. And so this will play into the icefish and what you saw happened with the icefish hemoglobin gene and why they ended up with clear blood. So bioinformatics is the use of computer software and math models to process and integrate data from sequencing projects. Okay. And what is a sequencing project? A sequencing project is basically figuring out, so when we looked at like DNA code and all the bases, it's basically mapping out every single DNA base and every single protein in an organism. And then the Human Genome Project was actually completed in 2003. I graduated from high school in 2002, so when I was in biology in high school, they were still like working on the Human Genome Project, so that was kind of a big deal. And so the purpose of the Human Genome Project was to sequence the entire human genome, and it has been done. Yeah, every year I'd say this. In 2016, whenever I got the numbers, obviously 5 years later, I probably need to update that. And I will do that, and I'm going to put a little note on here. I will do that whenever I go back and I make the Edpuzzle. But at the point, at 2016, which was 5 years ago, the genomes that were sequenced so far were 58,000 prokaryotes, 2,700 eukaryotes, and 5,300 viruses. So what I want you just to notice from these numbers is we know the genomes of more prokaryotes than we do anything else. Why is that? Well, there's more prokaryotes on Earth than there are eukaryotes, but it's because their genomes are smaller. So you all know eukaryote genomes are a lot larger and a lot more complicated. And so there will be less eukaryotes that have been sequenced so far. This is how it's done. So how do you think these genes were sequenced? And so basically, you don't really have to, you really don't have to write anything down for this, because this is it's kind of a complicated, complicated process. But basically, they're making linkage maps, physical maps, and then did DNA sequencing. And just write down a very complicated process using biotechnology that we actually do not cover in this class. Okay. And so what we can do with sequencing projects is we can compare. This is the basis of studying evolution, is comparing genomes and finding similarities and differences. So we can compare, and these are the 3 domains: bacteria, archaea, and eukaryotes. Those are the 3 domains, all right? And we talk a little bit about classification later. We'll talk about a little bit more about domains and kingdom, phylum, class, order, family, genus, species. I'm sure you probably learned that when you were in 7th grade, but that kind of plays a part in this too, although it's not as cut and dry as we want it to be. But I'll get to that a little bit later. All I really want you to notice from this chart is just to kind of draw some conclusions, okay? The conclusions that I would like for you to draw, or I'm just going to tell you what conclusions to draw, because if you looked at these numbers, I'm sure you guys would come up with this. Eukaryotes have a much larger genome than prokaryotes, than bacteria and archaea. So that's the first thing you want to write, is eukaryotes have a larger genome. The other thing that I want you to notice here, and it's kind of weird, and it's something that I just really can't explain, is rice and corn. Rice and corn are kind of interesting because if you notice, they have the most genes. And so something that I want you to just kind of know going into this is that plants are a lot more complicated than we know, and plants actually have more genes than animals do. So just kind of a cool little thing to know. So those are the only conclusions. Eukaryotes have a larger genome than the other 2, and then plants really have the largest number of genes compared to animals. Okay, and so let's talk a little bit about human DNA. 3 billion base pairs, 20,000 genes. What is all of that DNA like really made up of? And so something that's really interesting is that only 1.5% of our DNA actually codes for proteins. Only 1.5%. That's crazy. That means that 98.5% of our DNA is like useless, which is really weird, I think. So we have what's called repetitive DNA sequences, repetitive sequences. And so if you made a pie chart of human DNA, look right here where my cursor is. Exons. Remember, exons is what's actually cut out of the mRNA sequence and used for translation. Exons are 1.5%, introns are 5%. Then you have regulatory sequences. Regulatory sequences are going to basically regulate replication, transcription, and transcription. Unique non-coding DNA, also known as junk DNA, repetitive DNA, and more repetitive DNA. And I'm going to talk about what transposable elements are in just a minute. So we have just these repetitive, almost useless sequences of DNA that makes up most of our genome, which is kind of crazy. All right. We're going to skip that. Just go ahead and cross that out. Don't worry about that, because I want to talk a little bit about transposable elements. And so just kind of some questions. So you can cross out, what are SNPs, small nuclear polymorphisms, just cross that out. Why do you think there's so much non-coding DNA? I'm going to tell you. And how does non-coding DNA relate to evolution? I'm going to tell you. So non-coding DNA is basically going to give like a platform or a foundation for something to occur. And so I'm going to talk a little bit about what transposable elements are and how some of that non-coding DNA came to be. So transposable elements make up 75% of human repetitive DNA. These are stretches of DNA that can be moved from one location to another in a genome. Barbara McClintock is the scientist who discovered transposable elements, and she did that by studying corn breeding. And so there's two types of transposable elements. So remember, transposable elements are stretches of DNA that can be moved from one location to another. You have what are called transposons, and then you have what are called retrotransposons. So I'm flipping the page. Transposons and retrotransposons. So transposons can move within a genome via a DNA intermediate. And so what I want you to think about with transposons is it is either a cut and paste or a copy and paste mechanism that requires an enzyme called transposase. So here's a stretch of DNA. A transposon is just a segment of DNA that either gets cut and inserted into a different location, or it gets copied and inserted into a different location. And you're like, cool, what's the point? I don't really have an answer to that. I don't know what the point of that is. I don't know why that happens, but I do know that if DNA sequences get changed through this process, it could lead to coding DNA that codes for proteins that are beneficial to survival. I do know that. So this does play a part in the theory of evolution, because evolution is really based on DNA changes leading to more beneficial survival traits, right? So hopefully this kind of makes sense a little bit. It's weird. It's something that isn't really talked about that much because nobody really knows why it happens, but you can kind of think about if DNA segments are being cut and pasted or copied and pasted into different locations, that just leads to kind of by random chance, a new protein can form. A retrotransposon, exact same concept. A segment of DNA is basically being inserted into a new location, but it's going to go through the process of reverse transcription. And so what I really hope that some of you are seeing here is, wait a second, I've seen this before. This is kind of what HIV does. So the question that should be popping into your mind is, was HIV, the virus, formed through this process? And I don't know the answer to that. Were retroviruses here first and we kind of evolved from them, or did they evolve from us? I believe that viruses actually, that DNA and the organism was here first, the living organism was here first, and that viruses actually formed from this process right here. So a segment of DNA gets transcribed into RNA, then reverse transcribed back into DNA, and then inserted into a new location. Why does this happen? I don't know. And I'm going to keep saying that over and over again. There's so many missing pieces when we talk about the study of evolution, just so many missing pieces, but we do know that this happens. Okay, and then just kind of the same thing with transposons. It's going to kind of set the stage for new proteins and new traits to come into existence. So kind of cool. So what they can do is they can interrupt. So transposons and retrotransposons can interrupt or alter gene function. And so if you were really paying close attention to that video I just showed you, this is most likely what knocked out the hemoglobin gene for the icefish. Knocked out the hemoglobin gene. And then multiple copies of genes can occur. A lot of duplication, genes with related functions. Genes can diverge. Genes can become non-functional. When they're non-functional genes, they're called pseudogenes. and then eventually new genes with new functions can occur. So this is the why, or this is the effect, not really the why it happens. But this is the effect of transposons and retrotransposons. All right, so go ahead and pause if you still need to write, because I'm going to move on. What proof do we have? So if we believe transposons and retrotransposons can lead to these effects right here, what other proof do we have besides corn? We have what's called multi-gene families. Multi-gene families are a collection of 2 or more identical or really similar genes, and so we see that with hemoglobin. So there's different types of hemoglobin. It's believed that they all arose by transposons and the multiple copies of genes kind of arising. And so this is kind of a really complicated picture right here, but all I really want you to notice is that an embryo has a certain type of hemoglobin, a fetus has a certain type of hemoglobin, and an adult has a certain type of hemoglobin. So you have alpha globin gene families and beta globin gene families, and so these are just really similar genes with similar functions that are thought to have arose through the process of transposons, basically. And so on your notes, you'll see the question, other multi-gene families. An example of another multi-gene family would be antibodies. Remember, antibodies are proteins of the immune system released by B cells. And they're like the Y-shaped proteins that are going to surround a pathogen and render it inactive. So that would be another example. So I'm going to switch over for talking about transposons to something called transpositions. This would be a complete chromosomal rearrangement. So you can see how this is kind of a bigger picture. Transposons are a little bit smaller, just small segments of DNAs that get copied and pasted or cut and paste. Transpositions would be a complete chromosomal rearrangement. All right. And so look at this picture. Just kind of look at it. These are chromosomes. You have human chromosome number 2. All right, you see the telomere sequences at the end, and you're like, yeah, I remember what that is. There's centromeres, and there's a telomere-like sequence in the middle. Huh, what? Why would there be telomere-like sequences in the middle of a human chromosome? Well, let me tell you. At one point, these were 2 separate chromosomes. And what's the proof? Well, the proof is that the telomere-like sequences are in the middle. There's no, I mean, like, you know, I don't think there's any other explanation for that. The other proof or the other evidence that's used, chimpanzee chromosome number 12 and chimpanzee chromosome number 13, if you fused them together, they would be almost identical to human chromosome number 2. So what's the conclusion? The conclusion is humans and chimpanzees share a common ancestor. And in the common ancestor, there were 2 separate chromosomes that fused together, and then you had the evolution of humans. Okay? So chromosomes can break, chromosomes can fuse together. All right? The other thing I want to point out in this picture is human chromosome number 16. If you took mouse chromosomes 7, 8, 16, and 17, fused all those together, they would look very similar to human chromosome 16. So transpositions would just be chromosomal rearrangements through the process of evolution that can lead to a new species, basically. So what this is showing is humans and chimpanzees shared a common ancestor, and through the process of evolution, chromosomes fused together, and then humans evolved. And then on this picture right here, it's just showing that through the process of evolution, chromosomes fused together, chromosomes broke apart, mouse and human then evolved in their own direction. I know it sounds kind of crazy. Sometimes when I say it out loud, I'm like, do students actually believe me when I tell them these things? Like I said, there's still a lot of missing pieces. The next chapter, I am going to go through all of the evidences for evolution. So my job in this chapter and the next one is to try to persuade you to believe this. All right. So transposable elements, transpositions, are going to promote recombination. And I already said this, but I'm just saying it again. Disrupt genes, carry genes to new locations, they can be harmful or lethal. I would say most of the time, if a transposable element disrupts a gene, it's probably going to be lethal. But every once in a while, it could have a beneficial effect, and that can lead to the process of evolution. It provides the raw material for natural selection. Natural selection I am going to talk about in the next chapter. And this is usually where I show the film, but I wanted to catch your attention from the beginning. So the film about the ice fish, it is believed that through transposable elements, ice fish lost hemoglobin, gained antifreeze proteins, lost their scales. And that led to the increased in survival in sub-zero temperatures. And you can write down whatever you think is most interesting. And then just the climate change thing. I was just like being silly when I wrote this question down here. How do you think the climate change thing is going to play out? How is climate change related to the study of evolution? So climate change is a hot topic, obviously. How do you think it's going to play out? Well, I can tell you right now, the random mutations, the random changes in genes that occur in an organism, it happens by chance. If there are random changes in genes in organisms that lead to increased survival in hotter temperatures and more UV radiation, the more likely that organism is going to survive. So in humans, I think over long term, in order for the human species to survive all of this, is mutations that allow us to survive hotter temperatures and UV radiation are going to have to come about, or we'll have to do something for that. So anyways, I will talk more about climate change when we get into ecology, but evolution relates to climate change because species are going to be forced to evolve. There's going to be selective pressures on organisms where those who have the genes that are able to help them survive climate change are going to be the ones that survive it. Okay. And we already talked about that. That's what the video was about. New vocab word, it's called evolutionary development, Evo-Devo. And so you're just going to write down the definition of Evo-Devo. This is just the branch of biology that basically compares the developmental processes to understand how changes lead to evolution of organisms. And so pause and write that down, because then I want to say something real quick, and we're actually almost done. We're on the last page here. So pause and write that down. You see two pictures right here. This is called a phylogenetic tree. And you don't have to write that down, but I just want to say it so that you remember that I said it. When we get to chapter 20, we will start working with phylogenetic trees. I will have you make phylogenetic trees. Phylogenetic trees are just guesses, basically, of how things evolved over time and how closely related, evolutionary speaking, two organisms are to each other. So what I want you to notice here, time proceeds from left to right, kind of like the way you read a sentence. A branch point is going to be the common ancestor. And you're like, well, what is the common ancestor? Well, we don't know. We have no idea. We just think there was this common ancestor, have no idea what they look like or what they were, but it was an ancestor and it was common. And then over the point in time, things evolved in their own direction. So there was a common ancestor, bacteria evolved first, eukarya and archaea then evolved. What I want you to see here is, if a branch point between two different living things happened more recent in time, those two things are more closely related. So what you can conclude from this chart right here is eukaryotes and archaea are more closely related to each other than archaea are related to bacteria. And you're like, what? It's true. It's so true. All right. We'll get into that a little bit more later. This one down here, though, you'll probably understand a little bit better. There was a common ancestor. Over the course of time, the mouse, the chimpanzee, and the human each evolved into their own distinct species. The human and chimpanzee have another common ancestor that they share more recently. Therefore, out of these three organisms, the chimpanzee and the human are more closely related than the chimpanzee to the mouse or the human and the mouse. stay tuned. We will do more of that in chapter 20. Next thing I want to jump to is just, I have to mention the homeotic gene, because the homeotic gene and what's called the homeobox are used as evidence for evolution. So remember, we talked about the homeotic gene. I showed you the picture of the fruit fly that had like the leg coming out of its head. Homeotic genes are master regulatory genes that control placement and spatial organization of body parts. Okay. The homeobox is a 180 nucleotide sequence found within the homeotic gene. Okay. So the homeotic genes are thousands of nucleotides long. The homeobox is a small segment of the homeotic gene that is widely conserved. So what I need you to know, the words widely conserved, that means that it has stayed the same in all of these groups of organisms, fungi, animals, and plants. If a certain gene or if a certain DNA sequence is widely conserved, that is going to be used as evidence that we are all related through the process of evolution and that we all evolved from one thing. All right. Kind of a crazy concept, I know. So if you have any questions over that, let me know. And then this picture right here just shows you chromosomes. And it's just showing you fruit flies and mice, how that homeotic gene is very similar between fruit flies and mice. Therefore, at some point in time, the fruit fly and the mouse must share some common ancestor. All right, so it's used as evidence. I have not found this to be an issue at St. Mary's Hall, but I have at other schools that I've taught at, that certain religions teach against evolution, or certain religions say evolution's not real, blah, blah, blah. So basically, if we were in class, we would just have a little bit of a discussion of, if you had someone in AP Bio who was a friend of yours who was struggling because of their religious faith, what would you tell them? I have had students basically would not take notes, would not study, would not do anything because they thought they were going against their faith. It was really one student. She ended up failing the test. She ended up doing fine in the class. She got a 4 on the AP exam, but just couldn't bring herself to even listen during class. Like, you should have seen the look on her face every time I talked about evolution. So I would just say that this is the overarching theme of this class. And if anyone is struggling with it, just pretend. I mean, just because no one really knows. Well, I mean, I guess someone could argue with me, but that they think they know, but no one really knows. And this is it's a cool story. It's a cool topic. And we do have 6 whole chapters over this topic, and I've just covered 1. This unit is going to have 3. So if you are struggling with the topic of evolution, let me know and I can help talk you through it. But it is a huge topic in this class. All right. With that, I am going to end. And let me know if you have any questions about anything that I discussed. And then we are going to move on and talk about Darwin next time we meet. All right. Talk to you later.